In this video, we'll discuss the mechanism of action of ivermectin and its analogs. Ivermectin acts by interfering with the nerve and muscle functions of helminths and insects. In this picture, you can see the cross-section of a very common human helminth called Ascaris lumbricoides. It consists of body cavity, intestine with epithelial cells that absorb glucose, amino acids, and other nutrients, muscle cells that contract and move the pericytes body, nerve cells that innervate these muscle cells, oviduct, and uterus. This green rectangle represents the nerve cell or the muscle cell of the helminth. The drug binds to glutamate-gated chloride channels common to invertebrate nerve and muscle cells. Ivermectin can also bind to gamma-aminobutyric acid receptors. The binding opens the channels, which increases the flow of chloride ions inside the cell. You can see that the concentration of chloride ions inside the cell is now increased. The inside of the cell becomes more negative. This is called hyperpolarization. This excessive hyperpolarization is responsible for paralyzing and killing the helminth. Ivermectin belongs to the group of Avermectins. Avermectins are a group of drugs that includes ivermectin, selamectin, doramectin, eprinomectin, and abamectin. They have the same mechanisms of action as ivermectin. Ivermectin can be given by mouth, topically, or via injection. To understand why ivermectin is relatively safe for mammals, meaning it doesn't paralyze and kill us too, we need to understand the concept and the structure of the blood-brain barrier. The blood-brain barrier is the barrier between the cerebral capillary blood and the interstitial fluid of the brain. The blood-brain barrier is made up of capillary endothelial cells, with tight junctions between them. The basement membrane, pericytes embedded in the capillary basement membrane, and the foot processes of astrocytes. Now, I'll highlight the blood-brain barrier with this black rectangle. It acts as a barrier between the bloodstream and the extracellular space of the brain, allowing only certain substances like water, oxygen, and small lipid-soluble substances to easily cross from the blood into the brain. This prevents toxins, pathogens, and other potentially dangerous substances, like ivermectin for example, from crossing from the circulatory system into the brain. Ivermectin is safe for mammals, at the normal therapeutic doses used to cure parasite infections, because mammalian glutamate-gated chloride channels only occur on the neurons located in the brain and spinal cord, in other words, on the neurons from the CNS. Ivermectin and avermectins usually do not cross the blood-brain barrier due to the presence of the P-glycoprotein. Now, let's zoom in on this portion. Here you can see the P-glycoprotein located on the endothelial cells of the blood-brain barrier. The P-glycoprotein is one of the drug transporters that determine the uptake and efflux of a range of drugs. Ivermectin will get into the cell. The P-glycoprotein pumps chemicals and possible toxins from the cell. In our case, the P-glycoprotein will pump the ivermectin and avermectins out of the cell, preventing their passage across the blood-brain barrier. As a consequence, ivermectin and avermectins will not reach the receptors located on the neurons in the CNS. That's why ivermectin is safe for mammals at therapeutic doses. Now let's zoom back in. I want to show you what happens at higher doses. If ivermectin and avermectins are given at high doses, the P-glycoprotein will not pump the ivermectin and avermectins out of the cell quickly enough. Crossing may become significant, in which case brain levels peak 2 to 5 hours after administration, and it can be very toxic. So be careful with the dose. Different people can have different tolerance to the same dose of ivermectin, and I'll explain why. Let's zoom in on one endothelial cell. Inside the nucleus, we have DNA. There is a specific portion of DNA that goes through transcription, then translation, as a result of which the cell synthesizes the P-glycoprotein. This specific portion of DNA encodes for the P-glycoprotein, and is called the MDR1 gene. If this gene is normal, then the cell will synthesize a completely functional P-glycoprotein. And you know what the function of the P-glycoprotein is. The P-glycoprotein pumps chemicals, like ivermectin, and possible toxins from the cell, preventing their passage across the blood-brain barrier. Now imagine what will happen if a person, or an animal, has a mutation in the MDR1 gene. The P-glycoprotein that is synthesized will be abnormal, and it won't be able to pump ivermectin from the cell in sufficient amounts. A lot of ivermectin molecules will pass across the blood-brain barrier. These molecules will bind to receptors on the neurons in the CNS, and it will be very toxic for that person or animal. Ivermectin can be used against helminths and insects. Some examples of infections caused by insects, which can be treated with ivermectin are head lice infestation, caused by the head louse; pediculosis pubis, caused by the pubic louse; and scabies, caused by a mite called Sarcoptes scabii, which is an arthropod, specifically an arachnid. Helminths are classified into roundworms, also known as nematodes, and flatworms. Flatworms are further classified into trematodes, also known as flukes, and tapeworms, also known as cestodes. Some examples of infections caused by helminths, which can be treated with ivermectin are ascariasis, caused by Ascaris lumbricoides; strongyloidiasis, caused by Strongyloides stercoralis; trichuriasis, caused by Trichuris trichura; river blindness, caused by Onchocerca volvulus; and lymphatic filariasis, which can cause elephantiasis. Lymphatic filariasis is caused by filarial worms, which are highly specialized parasitic nematodes, for example Wuchereria bancrofti.